and thank you everyone for joining us. My name is Harry, and I'm a product specialist in microbial products at Norwegian. And the topic of today's webinar is a beginner's guide to 16S, 18S, or ITS Amplicon Metagenomic Sequencing versus the shotgun metagenomic sequencing. And I would like to break today's presentation into 4 parts. First 2 sections are introductions of Amplicon metagenomic sequencing and shotgun metagenomic sequencing. And then is the applications and a regarding case study. And after that, we will have a light discussion on other sequencing approaches in microbial research. And let's move to the first part, the introduction to Amplicon metagenomic sequencing. So first, let's go back to the last century, 1991, when environmental genomics was firstly brought to the world by Dr. Pace. It is based on the sequencing of amplicons, or we can call it PCR products, of the ribosomal rRNA genes. And the major purpose of Amplicon metagenomic sequencing is to reflect the composition of microbial diversity in each system. And in most of the cases, the Amplicon sequencing can be used to study diversity of bacterium, fungi, and some eukaryotic microbes in the community by applying the deep sequencing of the target regions listed here. And in 16S, 18S, or IDS Amplicon metagenomic sequencing, the target rDNAs, as is shown here on this slide, are composed of conservative regions, which are labeled gray. And they are interspaced by the hypervariable regions, which are labeled blue and green here. And those hypervariable regions can provide species-specific signature sequences, which will be useful for the identification of bacteria or fungi. For example, the V4 region of 16S rDNA is the most common target region for sequencing in order to identify bacteria. And what we usually do in Amplicon sequencing is that we use the universal primers that have the binding sites on the conservative regions to amplify the target hypervariable regions out. Then we will do the sequencing and conduct bioinformatic analysis to identify the phylogeny. So after some basic introduction, let's take a look at the project workflow of the Amplicon sequencing. So at Norwegian, we can provide the entire solution for 16S, 18S, or ITS Amplicon metagenomic sequencing, which means you give us your samples and we will return with sequencing data and bioinformatic analysis results. So when your first sample arrives, the lab technicians will perform the DNA extraction by using the different kits or methods based on the type of your samples. Then after the sample QC, we will use specific primers to amplify the target regions of interest and do purification by using the magnetic beads. And after that, the qubit fluorometer will help to quantify the PCR products and the agarose gel electrophoresis to separate and recover the target fragments. Then the next step will be the library preparation. And finally, we take the library to Illumina Nova-6 sequencer for the pair and 250 BP sequencing. So for researchers who would do extractions on their own, we have also several kits and method recommendations. So first ones are the classic surfactants and ethanol precipitation approaches like the CTAB and SDS. And also we could replace the ethanol precipitation with the magnetic silica beads. And the last one, also this kit, catalog number 51704 from Kaizen is also a great option for doing extraction of DNA from the saline soil samples. And after DNA extraction, we will move to the sample quality control. So for sample QC, we have mainly 3 criteria. First one, we could look at the picture to the left here. in which the main band is tight and can be clearly observed, and there is no obvious degradation. The criteria number 2 is that the DNA should be free of protein contamination. We can look at the picture here to the right that we can see bands here in the loading well, which is basically the protein contamination. And our third criteria is about the transparency of the samples. We should observe no obvious color, and the samples should be totally transparent. For example, we can see yellow and red in samples in this picture number 2 and 3, which are samples of abnormal transparency. This indicates that there might be some problem with the sample that could probably sabotage the subsequent processes. So in this case, we will continue the process only after we got confirmation from our clients. And generally speaking, however, we do not recommend to continue. Instead, we would suggest to redo the extraction for better experimental results. And after sample QC, we will move to the PCR amplification and the library preparation. So first, when we got the extracted genomic DNA, we will fragmentize them, then perform the end repair, the phosphorylation, the A-tailing, and then we will add the specific primers, which are linked with different barcodes to go PCR amplification. And after PCR is done, we will use qubit fluorometer for the quantification and agarose gel electrophoresis to recover the target amplicon fragments to move on. And in the next step, we ligate the amplicons with the indexed Illumina adapters to go to sequencing. So NOVA 6000 is used for the amplicon sequencing, and the strategy we apply is the paired end 250 BP. Oh, sorry. After sequencing, we can move to the data quality control. Here, we can label E as the sequencing error rate. So then the Q thread, which is the thread quality, can be written as the -10 multiplied by logarithm of E with base 10. So if the thread quality or thread score is 30, which means the error rate is 1 of 1,000, then the correct rate will be 99.9%. We call this Q30. And although the sequencing quality will drop during the sequencing, or the error rate will increase due to the consumption of the sequencing reagents, we are still able to say that in most of the cases, more than 75% of the reads could reach Q30, which exceeds the data quality of Illumina's official guarantee. So for data quality control, one of the important steps is the reads filtering. What we do is that we filter out 3 kinds of reads. The first one is that reads contains the whole or part of the adapters. And the second is the reads that contains more than 10% of the bases that are uncertain after sequencing. And the third is that the reads that more than 50% of the bases with thread quality score less than 5. So after this filtration, we will get clean reads for the subsequent analysis. And acknowledging all read sequences are stored in the FASTQ format. And we will include this pie chart in the data QC report, which shows the percentage of different kind of reads, as I have mentioned here. And after data QC, so if you are interested in the bioinformatics analysis, we will continue to the analysis part. So firstly, using the clean reads obtained, we will do the OTU clustering to categorize reads into different OTUs. And then the species annotation, the alpha diversity, which is the species diversity within samples, and beta diversity, which is the diversity among different samples, and all of those analyses will be conducted. And aside from the analysis items shown in this pipeline, we are also able to do a lot more advanced analysis, like the variance statistics, the correlation analysis, and the functional predictions using the clean reads we just got. And next, I will walk you through the part of the analysis individually. So first and the most essential one in the OTU identification. So OTU, namely the operational taxonomic unit, is determined with a cutoff value of similarity by the sequences of rDNA we got. And generally, this cutoff value is set at 97%, which means if the reads share 97% of the sequence similarity, they will be considered from one taxonomic level. So in the process of constructing OTUs, the basic information of different samples will be collected, such as the tags data. So tags basically are the reads that went through splicing after sequencing. And the summarization is shown at this figure. And in order to quickly and intuitively display the microbial community composition and the abundance information in the samples, we could construct a taxonomic annotation corresponding to OTUs. And the results could be viewed in this heat map. So from this heat map in the filter here above, we could input the count value per OTU. So when this value is decided and we keep the filter value unchanged, we can click this sample ID button. Then the graphic will be generated as the picture shows here. So it will show the different taxonomy and sample names and the corresponding OTU ID to the right. And the counts are colored based on the contribution percentage of each OTU to the total OTU count in one sample. The blue box means that it contributes low percentage of OTUs to a sample, and the red means it contributes high percentage of OTUs. And according to the analysis result of OTU clustering and the research requirements, we will normalize the OTU table and analyze both the common and the unique information for different samples or group. Then we will generate the vein or flower diagram. So each circle or petal here represents one sample or one group, and values in this overlapping parts represent the common OTUs, and the other parts are the specific OTUs in each sample. And for the sake of artistic, it is suggested to use the vein diagram if sample or group number is less than 5. And if we have more than 5 groups or samples, we can use the flower diagram. It will be much clearer to see. And then we can show the top 10 genera in high relative abundance to make the taxonomy tree by the independent R&D software. Now we can do taxonomy tree either in samples or in groups. So in this taxonomy tree, the different colors here represent the different taxonomic ranks, and the size of the circles are the relative abundance of the species. So the bigger the size, the higher the abundance. And the first number here below the taxonomic names represents the percentage in the whole taxon, while the second number represents the percentage in the selected taxon. And the Corona display is an interactive metagenomic visualization in a web browser. And Corona will visually display the analysis results of the taxonomic annotation. The circles from inside to outside stand for the different taxonomic ranks, and the area of the sectors means the respective proportions of different OTU annotation results. And also, according to the taxonomic annotation results, we select the top 10 taxa of each sample or group at each taxonomic rank, that is phylum, class, order, family, or genus, to form the distribution histogram of the relative abundance of the taxa. So we could visually see the taxa with higher relative abundance and their proportion in different classification levels of each sample. And the relative abundance of taxa in the phylum here is illustrated in these figures above. And also, according to the abundance information of the top 35 genus of all samples, the heat map can be drawn here, so as to check whether the samples with similar processing are clustered or not. And the similarity and the difference of samples can also be observed by this heat map. So plotted by the sample name on the x-axis, and the y-axis represents the genus. So the absolute value of z here represents the distance between the raw score and the mean of the standard deviation. So in this map, the redder the color, the more this phylum clustered in this sample, and vice versa. And to find the differences in the dominant taxa among the three groups or samples at the selected taxonomic rank, we can use the top 7 taxa to generate this ternary plot on the left. The three vertices represent the three samples or groups. So the circles here represent dominant taxa, and the size of circles are proportional to the relative abundance. So the higher the relative abundance, the bigger the circle. And the sample or group which the circle close to has higher abundance of this phylum. And also to further study the phylogenetic relationship of genus, we can choose the top 100 genera selected, and the evolutionary tree here is drawn using the aligned representative sequences. So this relative abundance of each genus was also displayed along the genus in this figure. And different colors of the branches here represent the different phyla, and different colors represent here in the different groups. And the relative abundance of each genus in each group was also displayed outside the circle. And alpha diversity is applied to the analysis of microbial community diversity within the sample by analyzing the diversity of the single sample, and we can reflect the richness and the diversity of the microbial communities in each sample, including the biodiversity curves and a series of statistical analysis. So generally speaking, the OTUs generated at 97% sequence identity are considered to be homologous in species. So here we summarize the statistical indices of alpha diversity when the clustering threshold is 97%. And the rarefaction curves and the rank abundance curves are also widely used for indicating the biodiversity of the samples. The rarefaction curve is created by randomly selecting a certain amount of sequencing data from the samples, then counting the number of the species it represents. And in the rarefaction curves, each curve here represents a sample and can be colored and shaped by each sample name supplied in the mapping file. And the sequence number is on the x-axis, and the observed OTU number is on the y-axis. So the rarefaction curve can directly reflect the rationality of the sequencing data volume and indirectly reflect the richness of microbial community in the samples. So if the curve is steep, lots of the species remain to be discovered. So if the curve becomes flatter, that means a credible number of samples have been taken, that only the scarce species remain to be sampled. And the rank abundance curve is to sort the OTUs in the sample by the relative abundance from the largest to smallest to get the corresponding sorting number. And then use the OTU sorting number as the x-axis, the relative abundance in the OTUs as the y-axis. We can connect these points with polylines. We will get the rank abundance curve, which can directly reflect the richness and evenness of species in samples. So for the rank abundance curves, each curve represents a single sample, which can be colored and shaped by each sample name supplied in the mapping file. And in the horizontal direction, the richness of the species is reflected by the width of the curve. And the higher the richness of these species, the greater the span of the curve on the horizontal axis. And in the vertical direction, the smoothness of the curve will reflect the uniformity of the species in the samples. So it means the smoother the curve, the more uniform the distribution of the species. And the beta diversity represents the explicit comparison of microbial communities based on their composition. So to compare the microbial communities between each pair of community samples, a square matrix of distance or dissimilarity was calculated to reflect the dissimilarity between certain samples. And the data in this matrix can be visualized with principal coordinate analysis, the PCOA, or the principal component analysis, the PCA, the non-metric multi-dimensional scaling, the NMDS, and the unweighted pair group method with arithmetic means, the UPIGMA. And weighted unifrac distance and unweighted unifrac distance were also selected to measure the dissimilarity coefficient between pairwise samples, which is a widely used phylogenetic measurement method in our microbial community sequencing project. So the heat map here, based on the weighted unifrac and the unweighted unifrac distance, we can see here to the left. So each grid here represents a pairwise dissimilarity coefficient between pairwise samples, and weighted unifrac distance displayed above, and unweighted unifrac distance will display below. So the smaller the sample, which means the smaller the species diversity difference between the paired samples. And the principal coordinate analysis, the PCOA, is an ordination technique which picks up the main elements and structures from reduced multidimensional data of the eigenvalues and eigenvectors. The technique has the advantage over PCA that each ecological distance can be investigated. And weighted unifrac, also weighted and unweighted unifrac, are calculated to assist the PCOA analysis. So each point here represents a sample and plotted by the principal component on the x-axis and another principal component on the y-axis. And the points were colored by group. So the percentage on each axis indicates the contribution value to the discrepancy among samples. So the more similar the sample composition, the closer they will be to each other. And the non-metric multi-dimensional scaling analysis, the NMDS, is a ranking method that is applicable to the ecological researchers. And it's a nonlinear model designed for better representation of the nonlinear biological data structure, which aims at overcoming the flaws in the method based on the linear model, including the PCOA and PCA. Okay, and next we will move to the introduction to shotgun metagenomic sequencing. So let's first take a look at what is shotgun metagenomic sequencing. And these two researchers, Kevin and Lior from University of California, Berkeley, they says shotgun metagenomics is the application of modern genomics technique, which does not need isolation and lab cultivation of the individual microbial species. So we know that, as we know that our biosphere has 10 to the power of 30 to 10 to the power of 31 microbial genomes, and only less than 1% of them could be cultured. We also know that during the isolation, there might be some species lost when applying different kinds of species enrichment. And when doing the cultivation, it is of high possibility that we would lead in bias due to the culture media preferences. So in this case, since it does not need cultivation and it will not lead in bias, by doing shotgun metagenomic sequencing, we will get a more authentic view of the microbial community structure. and how those microbes function within the community and the mechanisms and the metabolism network, as well as their interactions. And here is the common standard workflow of a shotgun metagenomic sequencing project. The first step is sample preparation. And often in metagenomic study, samples are from the target environment, such as water, feces, gut, or soil. And then DNA extraction is performed to isolate total DNA from those samples. And no gene can accept either environmental samples or DNA samples as input. And the next step after total DNA acquisition, we will apply the quantification and DNA quality control to make sure the DNA we have qualifies the standard of a successful library preparation. And then without amplification, the DNA will be fragmentized to about 250 to 300 base pairs for the construction of a 350 base pair insert library by a series of steps like end repair and adapter ligation. And finally, the qualified library will be put on the Illumina NovaSeq sequencer for data collection and the subsequent bioinformatic analysis. Let's jump into the workflow. And the first step, of course, is sample preparation. For metagenomic studies, we often take tissues or tissue adjacent samples, such as gut, rumen swabs taken from oral cavity or plant tissues to study endophytes. So in this case, there will be high possibility that the host DNA may be included in the total DNA extracted. So the host DNA sequences could take up a large portion of the total data obtained, and it will make the amount of the effective data very low for our subsequent analysis. So we should take measures to avoid the situation from happening. And we have some strategies to do that. The first one is that during sampling, try not to take the part close to the host tissue. And then when doing extraction, make sure to use the corresponding kit that is tailored for different kinds of samples. And also after sequencing, we could try to eliminate the host contamination by removing the data aligned to the host reference genome. And for those typical samples which researchers often focus on, we have several suggestions and tips regarding operation of sampling. So for soil samples, it is recommended to choose the soil which do not directly contact with air. And firstly, remove the visible impurities, such as the root of plants, tiny pebbles. And then we can pass the soil samples through a 2 milliliter diameter filter to further remove smaller impurities. For soil samples, it is recommended to collect samples from multi-spots of a location and mix them into one sample. And for gut samples, which is becoming more and more popular in recent studies, it is highly recommended to avoid touching the mucous membrane and make sure to wash the intestine several times with the PBS buffer. Then use a blade to gently scrape off the surface mucosa and collect them. This is a very crucial move to avoid host contamination for gut samples. And for sludge samples, if we can clearly see water in it, we would recommend to centrifuge the samples at 10,000 rounds per minute for 10 minutes at 4 degrees Celsius, and then remove the water, since in many cases, microbial diversity in water is quite different than that in sludge. And for swabs like oral cavity swabs, which are often collected now in this period of coronavirus pandemic, we would strongly stress to swab multiple times to collect sufficient amount of microorganisms. So commonly, we need 5 to 10 times of swabbing to meet the requirements of DNA amount in library preparation. And for water samples, if the waters were turbid with visible impurities, we would recommend to filter them using more than one specific diameters of filters. The ideal condition of this water is transparent with no visible impurities. And we know that replication is a precise process where the exact same findings are re-examined in the same way with identical design, which subject selection requirements and level of significance as the original research study. And the biological replicates are parallel measurements of biological distinct samples that capture randomly biological variation, which may itself be a subject of study or a source of noise. And for shotgun metagenomic sequencing, it is highly recommended to prepare at least 3 biological replicates for both environmental samples and plant or animal samples. And after we completed sample preparation, the next step will be the DNA extraction. Here we have some extraction methods recommendations regarding the different kinds of sample sources based on our project experiences. Also, one of the most extensively adopted extraction methods is using CTAB solution. So since the polysaccharides and DNA have different solubilities in CTAB, depending on the concentration of the sodium chloride, and at higher salt concentrations, the polysaccharides are soluble and which are insoluble, while at lower concentrations, DNA is insoluble. So consequently, by adjusting the salt concentration in lysate containing CTAB, the polysaccharides and the DNA can be differentially precipitated. And specifically for soil and sludge samples, this power soil DNA isolation kit from Mobile is very effective. And for stool samples, it is recommended to use the QIAGEN QIAMP DNA microbiome kit. And after extraction, the sample amount we need at Norwegian is more than 200 nanograms of total DNA, with concentration higher than 10 nanograms per microliter, and the total volume more than 20 microliters. After sample QC, we will move to the library preparation. So first, when we got the extracted genomic DNA, we will fragmentize them, perform and repair phosphorylation, A-tailing. In the next step, we ligate the fragment with indexed Illumina adapters without amplification. And also, NovaSeq sequencer is used for shotgun metagenomic sequencing, and the strategy we apply is pair and 150 base pairs. And after data QC, we will continue to the analysis part. So for analysis in shotgun metagenomic sequencing, we primarily would focus on the answers to these 2 questions. Who is there in our samples? That is the composition analysis, basically the taxonomic diversity and the phylogenetic diversity. And the other question is what are they doing? That how they are functioning in the sample's microbiota. And in our analysis pipeline, after quality control of raw data, we will obtain the clean data. The next step is by assembly of reads. The scaftics will be generated to do the following gene prediction. And after we get the non-redundant gene catalog, we will firstly perform the taxonomic annotation by using the NR database. And then we will do the functional annotation by using the CAC database to analyze biological pathways, and AgNoc database to do the orthology predictions, and KZ database to describe the families of structural related catalytic and carbohydrate binding molecules of the enzymes. And after we obtain those annotation results, we can further do pathway analysis, as well as some other clustering and dimension reduced analysis, such as the PCA, the PCOA, and MDS analysis I just mentioned in the Amplicon sequencing. So also metagenomics provides insight into the physiology of a community by clarifying the collective functions. And those functions are encoded in the genomes of the organisms that make up the community. And the functional diversity of a community can be quantified by annotating the metagenomic sequences by using the several databases here. So we have the CAC database, the eggnog, and the KZ. And for functional annotation graphs on this slide, the number on the bar here represents the number of unigenes on the node. And the x-axis is the code of each function class of level 1 in each database. So for example, on the CAC pathway annotation graph here to the left, the number on the different colored bars represents the number of unigenes annotated to the corresponding function. And the eggnog is actually similar to this one in CAC pathway annotation. And according to the annotation results of each database, the relative abundance statistics of the functions in each database can be drawn. So we take the relative abundance statistics of each sample at level 1 in each database as an example. The vertical axis here represents the relative proportion of annotations to a certain functional category. And the horizontal axis represents a different sample. Here is the sample name. And in the metabolic pathway diagram, the node here represents the various compounds. And the lines represents a series of enzyme reactions. The red color means the enzyme reaction shared by two groups or two samples. And the blue represents the unique enzymatic reaction of group A or sample A. And the green represents a different unique enzyme reaction in different group or in different samples. And whether it is human gut microbes or other environmental microbes, there are many resistant genes. The abuse of antibiotics leads to the irreversible changes in the microbial community, in the human body and the environment also, so which poses risks to human health and the ecological environment. So therefore, the research on resistant genes has received extensive attention from researchers. CARD, this comprehensive antibiotic resistance database, is a newly emerged resistant gene database. In recent years, it has the advantages of comprehensive information, the user friendliness and time update and the maintenance. So the core component of the database is the antibiotic resistant ontology, the ARO, which integrates information such as sequence, the antibiotic resistance, and the mechanisms of action, and associations between AROs, and provides the online interface between AROs and PDB or NCBI and other databases. So this bar plot to the left is the relative abundance of resistance genes like we have discussed before. And the donors chart to the right here is plotted according to the taxonomic annotation of all the samples in the group. And the proportion of different phyla in all genes and resistant genes are shown in this donor chart. So which phylum had more resistance genes can be visually reflected. The inner circle is the species distribution of AROs, and the outer circle is the species distribution of genes of all samples in the group. And the next part is the applications and the case study of Amplicon Metagenomics and shotgun metagenomics. So speaking of the applications of Amplicon sequencing or the shotgun metagenomics, I have listed different fields here. So in agriculture, the Amplicon and shotgun metagenomics are often used in the study of microbial species and their functions in plant root systems or different regional soils. Well, in the medicine field, they are often used in explaining the relationship between the species composition and functions of different flora and diseases in both human gut or the human oral cavity. In animal husbandry, we would focus on studying the microbial diversity and functions under the different nutrition status on their immunity levels. The samples are often from animal rumen or intestine. And also Amplicon and shotgun metagenomic sequencing can be applied in fermentation industry, the environmental governance, and the biofuel research in ocean and activated sludge. So far, we have finished introducing the analysis content and the application. So now I would like to share this interesting paper with you. This paper is published on Gut Microbes in 2020. It is about how spaceflight could influence the function and composition of human gut microbiota. And as we know, space is a dangerous environment and the risk to human health. And astronaut physiologies change profoundly when they exposed to the environment in space. And previous spaceflight studies have shown that the astronauts were easily infected by bacteria, viruses, and other pathogens because of the immune dysregulation up there in the space. So in order to study how will spaceflight influence composition of human gut microbiota, the authors collected fecal samples from astronauts from this who took spaceflight mission 1. There are 3 people, namely the S1, S1.2, and S1.03, and the second spaceflight mission. This is there are 2 people, the S2.01 and S2.02. So from the fecal samples, they extracted in the total DNA, and they constructed a 350 BP insert DNA library. Then the library was sequenced on a Illumina NovaSeq sequencer by using this pair and 150 BP strategy. So by doing this following analysis, they have some interesting findings that I would like to share with you. The fecal samples were collected from each person's at 4 or 5 time points, including 1 week before lunch, or 1 week before the launch of the space flight, which is marked L-7, and recovered 1 day after the space flight, that is R+1, and recovered 1 week after space flight, that is R+7, recovered 2 weeks after space flight, R+14, and recovered 4 weeks after the space flight, R+28. So the author first characterized the gut microbiota compositions of mission 1 at the phylum and the genus levels. It's in figure A and B. The gut microbiota is here at both the phylum and genus levels. We can see it's fluctuated obviously between R+1 and R+28. And then the author characterized the composition of the gut microbiota from mission 2 at phylum and genus levels, which is figure C and D. And the gut microbiota at both the phylum and genus levels were also significantly influenced by the spaceflight. And also the recent studies on microbiology in space have revealed that the spaceflight has an important influence on bacterial resistant genes. The results of this study showed that the only unique enzyme reactions affected by spaceflight were those involved in the biosynthesis antibiotics. So the author analyzed the resistance genes in the gut microbiota from mission 1 and mission 2. So here the left-C analysis was used to obtain the different AROs, and they compare the ARO abundance of R+1 with that of R+28 from mission 1. And the LDA score of the AROs above 2 and the cluster heat map are drawn here. And we can see that they have changed significantly. So from the graphs on these 2 slides, it is shown that the species abundance and resistance genes in the gut microbiotas were both influenced by spaceflight. This is a paper which used typical shotgun metagenomic sequencing approach that studied and reviewed how spaceflight could affect composition of human gut microbiota. And during communication with our customer, we have found that some of them may get confused of whether choosing amplicon sequencing or shotgun metagenomic to study the composition or function of microbiota. Here we have a slide showing some differences between them. So firstly, in amplicon sequencing, we will amplify a certain target region. For example, 16S-V4. This is often our region of interest in bacterial study, while in metagenomics, we do not do any amplification to genomic DNA. We just use the DNA directly to proceed to library preparation, so we can get a more authentic view of the samples. And secondly, the sequencing strategy of amplicon is paired at 250 BP. Some may use the pair at 300 BP, but for the metagenomics, is basically pair at 150 BP. And we need relatively smaller data amount in amplicons. Commonly, it 30K to 100K raw reads per sample. But for shotgun metagenomics, we will sequence a large amount of data. Normally, we will choose the 6 gigs. And for amplicon analysis, we focus on the microbial community structure or composition by doing the OTU clustering and the diversity analysis. While in shotgun metagenomics, although we could also study the composition, but what we care more is the genome and its functions by doing the gene prediction and the annotation based on various databases. And here I made a cartoon that shows the comparison among resequencing, shotgun metagenomics, and the Amplicon metagenomic sequencing, which are the three most common sequencing services you can find on the market. So after we done sampling from water or feces or soil or gut samples, if we want to study a single bacterium or fungus, we will do culture and separation. The most common goals are studying the genome composition, single nucleotide polymorphism, insertion or deletion, structure variation. And for the mutation researchers of known microbes, we often choose the microbial resequencing. And if you want to study the microbial diversity, at the same time, we care more on the metabolism pathways of genes, the gene functions. We just use total DNA after host depletion, of course, to do the shotgun metagenomic sequencing. But if all we care is the species identification and the species diversity, commonly we will choose the Amplicon sequencing, and it's the most cost-effective approach. So in the last part of this presentation, we will discuss a relatively novel sequencing approach to study microbial composition and diversity in your field of interest. And there are some times that we care more about the whole 16S rDNA than just a few hyperviable regions. So this often happens when our purpose is to study the gene distribution along the entire rDNA. So in this case, the long read length of Microbial platform can perfectly fit in. It is able to sequence up to 1,500 BP CCS reads without fragmentation of genomic DNA. Since in CCS, or we call it hi-fi mode, the Microbial sequencer will sequence a read for a great many times and pick the most accurate one after comparing them to each other. So the accuracy is very high, which can go up to 99.999%. That is the QV50. This is a major advantage when we want to identify or quantify some very rare variants. Also, the full-length 16S Amplicon in PacBio platform greatly improves the resolution, that is the percentage of sequences which are classifiable at species level. This is a statistical figure from a paper. The x-axis are the sample names, while the y-axis is the percentage of sequences classifiable at species level. It is very obvious that the percentage vary from 32 to 93% in Illumina platforms, depending on the hypervariable region. But PacBio exceeds totally it at most 100% in each samples tested. This means by using PacBio, we can get the potential to identify more species or biomarkers than by using the NGS platforms. So we are able to obtain a more authentic structure of the microbiota. And at Novogene, we also provide full-length 16S Amplicon sequencing service. So if you're interested, please feel free to ask our representatives for more information. And on this slide, we show the workflow and extraction kits recommended for doing the full-length 16S Amplicon sequencing. With all that being said, so what makes us different from other companies in the market? The most important one is that we have a vast majority of experiences in Amplicon and shotgun metagenomic sequencing. And our clients had published various papers on related journals, and up to the end of 2020, the total count of published papers by customers have reached 100, with a total IF of 620. And also, I'm very proud to say that Novogene has this Falcon, a fully automated intelligent platform that could integrate all pre-sequencing processes from extraction to library quality control. In this way, Falcon can greatly shorten the project turnaround time by processing 2,000 to 3,000 samples in just 24 hours, which could lead to a 60% decrease of production time and the turnaround time. And thanks to our world-leading computing and server capabilities, we could carry a vast number of projects in parallel, and at the same time, still achieve amazing data stability and turnaround time. And also, we are grateful to have more than 2,600 of collaborating research organizations and universities, more than 1,200 collaborating companies, and more than 590 hospitals all around the world. We are proud of so many partners working and doing research with us, and we hope you could be one too. So this brings us to the end of the presentation. Thank you for joining us. And also, I would like to thank our fantastic panelist group for supporting today's webinar. So if you would like more information, do not hesitate to see our website at en.novogene.com or send us email at inquiry_us@novogene.com. So for customers who speak Spanish or Portuguese, we have also a dedicated sales manager, Dr. Diego Levi, who is now based in Argentina. Please contact us for any information, and we are responsive and happy to help at any times. And I also noticed that there are questions raised in the chat box. Some of them were already answered by our panelists. We will answer the rest of the questions by email and send it to you afterwards. So thank you everybody for your time and attention.